Welcome, everyone, and thank you for joining us for today's webinar, the NIH Microphysiological Systems Program, presented by Dr. Danilo Tagle, an Associate Director for Special Initiatives Office of the Director of National Center for Advancing Translational Science, National Institute of Health. My name is Susie Valdez, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational and virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Just simply click on the Ask a Question box located on the far left of the screen and type your questions into the drop-down box. Our speaker will respond to you and your questions via email following the presentation. If you have any trouble hearing or seeing the presentation, just click on that Ask a Question box and let us know you're having and experiencing a problem. This presentation is educational and thus offers continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of your presentation window and follow the process to obtain your credits. Without further ado, please join me in welcoming our presenter today, Dr. Danilo Tagle. Welcome, Dr. Tagle.
Yes, thank you, Susie, for the introduction. I'd like to go over the NIH Microphysiological Systems and essentially provide a general and broad overview of the program and tell you what's currently going on in this program, and then also lead into future directions. So this will be my outline. I'll start with telling you a little bit about the National Center for Advancing Translational Sciences, where I'm Associate Director for Special Initiatives there. And as I've said, I'll work you through the Microphysiological Systems Program, tell you about the partnerships that we have, and then go into future directions. So the National Center for Advancing Translational Sciences was stood up at the NIH, the National Institutes of Health, back in 2012. And its central mission is to catalyze a generation of innovative methods and technologies, essentially to enhance the development of and implementation of diagnostics and various therapeutics. So you can tell right away that it's very different from the rest of NIH, which is focused on specific group disorders or organ systems. So what we're really about is to serve as a catalyst in terms of resource and tools development that would aid in terms of quick implementation of various therapeutics and diagnostics into the clinics. Now, if you're familiar with the drug development pipeline, there's certainly a number of stages from target development all the way to clinical trials. And currently the paradigm is to use a variety of tools and technologies that includes 2D cell cultures and in vivo animal models, and before getting into human trials. However, there are many significant challenges in drug development currently. The average length of time to develop and bring to market a particular drug is on the average from 10 to 15 years with a cost of $2.6 billion. And so the current drug discovery paradigm has a failure rate of 90%, 55% of which is due to lack of efficacy, and an additional 28% is due to toxic effects in humans. So what we need and what NCATS is trying to address is to create tools that would be better predictive of the human response to some of these candidate therapeutics. If you look at the safety and efficacy issues, a good number of the failures is because we are using currently animal models that are not predictive of the human response. One of the recent publications indicate that no matter what animal model you use, whether it be a dog model, even non-human primates, rodents, and this is a recent paper looking at 4,000, close to 4,000 drugs that have gone through FDA approval over the past 70 years, indicate that probably dogs would be at best predictive of about 25%, as well as strats for various human conditions. But generally, all these animal models are not necessarily useful in predicting human response. And that is where the microphysiological systems program or the tissue chips come into play. So essentially what we have are a multi-organ microfluidic device that contains human cells and tissues. So just to walk you through what a microphysiological systems look like, or tissue in a chip, you will have a scaffold, which means having the right extracellular matrix represented in the system, cell sources that are not monolayer 2D cultures, but essentially multicellular cell types that represents the tissue composition and representation of a particular tissue system or organ systems, and also coming from renewable cell sources such as stem cells, like induced pluripotent stem cells or primary cells. The structure, such as the rigidity and porosity of the particular tissue, is also represented in this system, as well as spatial and temporal patterning that includes cytokines, hormone gradients, even oxygenation gradients are also represented. Of course, since these are 3D cell culture system, perfusion is an important factor where nutrients, oxygen, and various pH are balanced and represent the actual physiological conditions. The bioreactors includes biomechanical transduction properties so that the actual tissue types undergo biomechanical stimulation. Innervation, immune host response are also critical features that we're trying to represent in the systems, as well as functional readouts that would include non-invasive real-time imaging in this system. So this is just a general representation of what a tissue chip look like. And I've said it's really trying to represent physiologically as well as in terms of the cytoarchitecture of particular organs and tissues, and using real-time imaging and omics-based technologies to have the capability of newer ways of evaluating response of these devices. So the NCATS tissue chip for drug screening program, just to reiterate, essentially has a central goal of creating these in vitro platforms that uses human cells and tissues. And its central goal is to represent at least 10 major organ systems: the circulatory, the endocrine system, the GI, the immune system, skin, musculoskeletal system, the nervous system, reproductive, respiratory, and urinary system, with the goal of making it physiologically relevant, genetically diverse, and of course needs to be pathologically meaningful. It's intended to be modular, that is, it can be reconfigured such that individual organ system can be plugged into other organ systems to form multi-organ, or to the point of actually creating a human body on a chip, and at least viability of the cells and tissues for at least 4 weeks. Just to give you an example of how organ function is represented in this organ system. So for example, if you look at the lung, you reduce that to the basic functional elements. So we're approaching it from a reductionist approach, where you look at the air sacs or the alveoli as the basic functional unit of a lung, and that is represented on the right-hand side of the alveoli as the device wherein cyclic vacuuming creates this negative pressure that allows a membrane containing, shown in blue, the lung epithelial cells, and below that endothelial capillary cells, which would undergo stretching, similar to what would happen in the alveoli. So the process of the stretch actually stimulates the lung cells to form surfactants and be much more responsive and representative of the actual condition in the air sac. And I'll show a demonstration of that later on. So there are a number of platforms, engineered platforms that are now represented in the NIH tissue chips program. So the one on the top left panel would be the actual tissue chip that is being used to represent the lung. And then the one in the middle is a microvasculature in the chip, where you can actually embed tissues and other tissue constructs, such as even tumor cells, to vascularize that particular tissue construct. The one on the right is a kidney on a chip, which contains a proximal tubule of the kidney. The bottom left would be an example of an integrated multi-organ system. So in this particular case, this is the female reproductive system that contains a fallopian tube on the chip, contains a uterus, ovary, the ectocervix, and also connected to the liver on a chip, which would then inform drug developers in terms of how hormonal changes, in this case a female cycle, can influence drug metabolism by hooking this up with the liver. And the one on the right is also another example of an integrated multi-organ system that contains the skin, the liver, heart, and bone in this particular case. So there's a wide variety of microengineered platforms, as well as the different ways that organs and tissues are represented in this system. Just to give an example of how these models are being used to predict, for example, efficacy. So here's an example where the lung on a chip is used to model pulmonary edema, where it's very hard, or if not difficult, to model this condition in any animal or even in vitro system. So the scenario is to look at IL-2 chemotherapy, which is used primarily as indication for kidney cancer and melanoma, but has the unfortunate side effect in a few instances of vascular leakage syndrome. So in the bottom panel is shown the lung on a chip, and wherein you're seeing FITC labeled inulin. You can see that if there is the labeled inulin will traverse through the capillary and lung epithelial cells, showing leakiness of the capillary bed and going into the lungs, which would be representative of pulmonary edema. However, if you then incorporate a TRPV4 inhibitor into the system, still with a 10% strain of stretching, you can see that leakiness, vascular leakiness, is mitigated in the presence of TRPV4. And so showing that this condition can essentially be prevented or can be sort of a companion drug to IL-2 chemotherapy. Another example that I'd like to show is the functional integration of various tissues on a chip system. So in this particular case, linking up the liver, the kidney proximal tubule with the GI system or the gut, along with the neurovascular unit or the blood-brain barrier, informing how multi-organ connectivity can influence or inform how drugs can be metabolized in the system. So an example of that would be looking at aristocolic acid, which uses nephropathy in a number of cases. So this is known to be ingested through Chinese herbs medicine, which primarily being used for a diet. It comes from the plant Aristolochia, and when ingested and have gone through liver metabolism, as shown in the picture, in the figure here, that in the presence of liver and kidney, the proximal tubule of the kidney shown in red, undergoes apoptosis. And that is shown in the diagram where just the kidney cells by itself treated with aristolochic acid are viable, whereas when the kidney proximal tubules are connected to the liver and the aristolochic acid undergoes metabolism, it converts aristolochic acid into its sulfated form, which is then nephrotoxic. And then the mechanism of action can also be shown in the sense that the sulfated conjugate form enters the kidney through the OAT transporters, which can be blocked by the treatment with probecenid, as shown in the bottom right-hand figure, where the treatment probecenid essentially renders the proximal tubule cells to be viable, and this is shown by staining for live dead cells in this particular case. Just to give you an overview of what has been going on in the first 5 years since the inception of this program. So the NIH has supported this program for the first 2 years to develop the platforms and as well as create the cell resources. And then the last 3 years, from 2013 to 2017, was essentially to validate the systems. That is, are these models actually representing the tissue and the organ physiology, and then using a training set of compounds that AstraZeneca, Pfizer, and GSK guided us in terms of what to use. So essentially, this training set of compounds are molecules or chemicals that have gone through preclinical assessment and has been approved by the FDA for human trials, but subsequently has been withdrawn because of adverse events. So the question would be that we're asking is, with this model system, the tissue chips, would the human response have been predicted ahead that the animal models and the in vitro 2D culture systems was not able to predict? And so that was sort of like the activities that have been going on in the last 3 years of the program from 2013 to 2017. We have also partnered with a number of government agencies such as DARPA from the Department of Defense, where their primary interest is in developing the 10-organ system or the human body on a chip. And then the FDA is a non-financial partner, but essentially providing NIH with key regulatory advice in terms of how to qualify and validate this system for regulatory decision-making. So out of the 5 years of the program, a number of publications have already come out, over 500 of them in various high-profile journals, in some ways informing the scientific community in terms of how this or various tissues on the chips can be and is being used in drug development. Now, just to give you the current landscape of what the NIH tissue chip program looks like, we now have formed a very active consortium made up of investigators, shown in the bottom right, the tissue chip developers with Tissue Chip 2.0 showing the various academic institutions wherein all these activities are going on, and NCATS essentially form a leadership role in providing support to make this technology move forward with the Tissue Chip 2.0 that is now actively moving forward from 2018 for the next 5 years to model diseases, various diseases, and I'll show you a little bit of what those diseases are. We also have a number of partnerships, both within the NIH, shown in the top left corner, also with a number of pharmaceutical companies through GSK, AstraZeneca, Pfizer, and Thermo Fisher, and also through the IQ Consortium, also with a number of government agencies such as the FDA, NASA, DARPA, and BARDA. What is interesting is in the is the support that NIH has continued to provide also to startups and spin-off companies that have come from the academic institutions that are now seeking to commercialize these model systems, and those are shown in yellow. A key outcrop of the partnerships that we have with the IQ Consortium and the FDA are the tissue chip testing centers and the tissue chip database center shown in green. And I'll tell you a little bit more about that in subsequent slides, and also tell you a little bit about our partnership with NASA and the Center for Advanced Science in Space, shown in red on the top right corner, in terms of how we are taking advantage of unique conditions in space, both to advance biology as well as the technology in this program. So for the disease models, which is the tissue chip 2.0, we're essentially looking at monogenic or polygenic diseases, in either Mendelian or multifactorial, and how this could be modeled in various tissue chips, whether through single organ or multi-system involvement for the disease pathology. And essentially what we are striving to do is to develop various readouts, not only using conventional staining and biochemical analysis or enzymatic analysis, but also using omics-based technologies, real-time imaging that are non-invasive, gene expression studies, which can then be used to inform and correlate with the in vivo human condition, looking at absorption, distribution, metabolism, excretion, concentration, and various toxicities, and then be able to do the in vitro and in vivo extrapolation studies to inform and continue to improve our model system. So in order to inform us how these devices, these microphysiological systems, can be used to model diseases, we first started with rare diseases as a way to work it into the system, because typically those are single Mendelian monogenetic traits. And so we have our tissue chip developers partner with another NCATS program, which supports the Rare Disease Clinical Research Consortium, shown here in red, or work directly with patient advocacy groups, shown in yellow. And so out of this activity, a number of rare diseases, especially vascular malformations, lysosomal storage disease, tuberous sclerosis, kidney disorders, liver disorders, and heart, and a number of neurological disorders as well has been modeled using the system, which then led into a full-blown program, which we're calling Tissue Chip 2.0, which is then focused more on disease modeling, and not just on rare diseases, but certainly on multifactorial and polygenic disorders. So shown here are the current efforts to use these tissue chips to model various diseases, from xerostomia, which is a drying of the salivary gland as a consequence of chemotherapy, which is a single organ modeling, to multiple organ involvement, such as in type 2 diabetes, shown in the bottom right, in the blue box, wherein various groups are modeling type 2 diabetes by connecting, for example, the pancreatic beta cell, islet cells, with the liver, with GI system, and with adipose tissue, to accurately depict what's going on in type 2 diabetes. So there are certainly a number of opportunities in applying tissue chips for rare disease research, and especially for using patient-derived iPS cells to populate these tissues. This can also be used to look at multi-organ involvement and many rare diseases, and then, of course, be able to evaluate without necessarily compromising the patient to evaluate multiple treatment approaches in parallel. So then it protects the patient naive status until the best treatment option is identified. So with this effort, one of the examples is looking at, for example, Bart syndrome, which is an X-linked recessive disorder caused by a mutation in the capasin gene and chromosome X. It is caused by a severe cardiolipin deficiency and essentially characterized by cardiomyopathy. And it's one of the rare diseases, about 1 in 300,000 births. And so this is how it's represented in a tissue on a chip, wherein cardiomyocytes from patients are put into muscular cardiomyocyte thin films, wherein the beating rate in terms of the rhythmicity of the cardiomyocytes beating is captured in the system as a sort of like a cantilever type device readout, where the beat rate is measured in terms of the deflection of the cardiomyocyte thin film. So in this particular case, taking patient-derived iPS cells and either also putting them on chips, and then also taking isogenic lines, iPS isogenic lines, and through genome editing, incorporating the same changes, the same mutations on those normal isogenic lines to make sure that the actual pathology is due to the mutation, and then being able to evaluate in this system various candidate therapeutics that could help mitigate some of the cardiomyopathies in this particular disease. So this is an example of the Bart syndrome on a chip, where the cardiomyocyte shown in figure A, that's the tissue chip with the microelectrode array. Figure B would be the sarcomere organization, shown in labeled with actinin, and then the figure C would be the top figure, would be a normal cardiomyocyte from a normal individual or a non-Bart syndrome patient. And then the one in the middle would be showing the cardiomyopathy from a Bart syndrome patient. And then the bottom would be treated with, in this particular case, treated with a cDNA that corrects the mutation in the iPS cardiomyocytes, showing restoration of the sarcomere structure. This is also shown in the beat rate. I guess the video is not showing, but essentially what you would have would be slow beat, the beating in the left-hand corner of D, and then the one in the middle would be cardiomyocytes from Bart patient that is showing the cardiomyopathy and it's not beating or hardly beating, and then restoration of the contractility of the Bart syndrome cardiomyocytes when treated with radical oxygen species scavengers or treated with a cDNA that restores function into these cardiomyocytes. What I'd like to show too, as I mentioned, that there are a number of startups and spin-off companies that have grown around this technology, and shown here are a number of those companies that are either commercializing the actual chips themselves or in the process of providing fee-for-service to test various therapeutics in the system. Next would be showing how pharma and the FDA is also showing a great interest in the use of organs on chips in terms of drug development and regulatory decision making. So for example, recently in 2017, the FDA has actually signed a collaboration with Emulate, essentially to bring Emulate technology. So Emulate is one of the companies, the startup companies that are seeking to commercialize the lung on a chip, the GI on a chip, the blood-brain barrier on a chip. And so the FDA is looking to set up and has set up a facility within the FDA campus using the Emulate technology. Likewise, they have also partnered with CN Bio, which is commercializing the liver on a chip, as well as a multi-organ platform. Likewise, to test that platform at the FDA. And then recently, the FDA also have a public hearing in terms of how this type of new technology, in vitro technology, can be used to predict human responses to a wide range of substances. In terms of pharma interest, Johnson & Johnson and Merck have also partnered with Emulate in terms of using this organ-on-chip technology in terms of their drug development pipeline. And similarly, AstraZeneca have also partnered with Emulate to set up a chip-based, tissue chip-based technology as part of their drug development pipeline in terms of their laboratories in Sweden. So our work with the pharma companies have largely been facilitated through the IQ Microphysiological Systems Affiliate, IQ Consortium. At this state consists of representation from about 20 pharma companies, all interested in engaging with tissue chip technology and looking at how this can be used to inform drug development by looking at drug metabolism and some of the safety and issues, looking at biodistribution of the drugs, and essentially because of this IQ consortium, member companies can essentially compare notes and be able to provide input to the NIH as well as to the FDA in terms of how to best deploy this technology in drug development. So part of the activities that we've had with the IQ consortium and the FDA is essentially to create a validation framework wherein this technology can be used to build confidence in how it can be effectively used in drug development. And so we view this as essentially three steps in terms of validation. One would be physiological, wherein the organ function and structure is validated and represented, and these are primarily done by tissue chip developers. And as I mentioned earlier, this is done using a training set of compounds that was informed by AstraZeneca, Pfizer, and GSK. We also have the second stage, which is the analytical validation, and this is largely conducted by independent testing centers, and essentially looking at technology transfer to these laboratories and being able to determine the reproducibility, the reliability, the robustness, and relevance of individual organs on chips, and how they can be used for just using a validation set of compounds that was guided by the IQ consortium at the FDA. And as I've said, this is done through what NCATS stood up as the tissue chip testing centers, where data is also collected through the tissue chip database centers. And lastly, the industrial validation stage is where industry and regulatory agencies would then start adoption and commercialization of this technology and started testing on them proprietary set of compounds, and largely done through a CRO type environment. So just to give you a little bit of flavor in terms of the tissue chip testing centers, so NCATS has established this as a resource center, funded initially for 2 years, starting in 2016. And we have recently renewed them for another 2 more years of funding, with a goal that after the after 2020 that they will be self-sustaining and being able to generate revenue coming in from their interactions with the various stakeholders in the field. As I mentioned, FDA and IQ Consortium was critical in providing guidance on the validation set of compounds, as well as the type of assays and biomarkers that will be used in terms of independent validation of each of the organ systems. So the testing centers that we at NCATS have funded is through MIT, Morat Sirit and Alan Grodzinski are the PIs, and then also Texas A&M University, where Ivan Rissin is the main PI. The database, the Microphysiological Systems database, is housed at the University of Pittsburgh, and the PI for that is Mark Sherda. And so on the bottom are the list of the various tissue on chips or organs on chips that have gone through initial testing at these testing centers and validation. And just recently, the first publication coming out of the testing centers was published in Nature Scientific Report, and that was done by the Kidney on a Chip. So the idea here is for each of the testing centers to test each of these organs on chips in parallel, essentially to look to be able to independently validate how useful the technology is, how easy it is to transfer the technology from the developer to the testing centers, and being able to replicate the findings through the validation set of compounds, creating best practice, and being able to identify what the primary opportunities are in using these technologies, as well as identifying some of the limitations. As I mentioned, we have renewed the testing centers for another round of funding with the goal of transitioning them into a self-sustaining model. So with the MIT Testing Center, which is transitioning into a CRO, Javelin Biosciences, and they have a business model of working with some of the startups and spin-off companies that are commercializing some of these technologies and being able to validate the systems as they come into market. The Texas A&M University has the business model of working with the tissue chip developers and looking at validating disease models, looking at some of the more improved tissues on chips, some of the more subsequent iterations and improvement, and also looking at how a number of the systems can be used for efficacy testing. One exciting thing that I'd like to indicate is the partnership that NCATS have with NASA and the Center for Advancement in Science and Space. And this is essentially looking at opportunities to advance organ-on-chip technologies, both in terms of looking at biology as well as technology improvement. So one of the main things that we're looking at would be some of the changes that happens under microgravity conditions in space. And so this is done with research at the International Space Station. So astronauts who have gone through prolonged stay at the International Space Station undergo a number of physiological changes. So within 3 weeks that they're up in space, they undergo upper body fluid shift, neurovestibular or inner ear changes, sleep disturbances, bone demineralization. From 3 weeks to 6 months, they also undergo bone resorption, muscle atrophy, vascular deconditioning, GI disturbances, hematological changes, and then anything more than 6 months, in addition to all of the above, they also have declining immunity, renal stone formation. All of these changes happens in a very short period of time. And the interesting observation is that when the astronauts return to Earth, there's a reversal to normal physiology within a few weeks to months when they return to Earth. And so this affords us an opportunity then to look at some of these changes and be able to figure out what instigate some of these changes under microgravity, determine what the molecular signatures are. And if you look at some of these physiological changes, these are essentially some of the presentations that would remind you of aging. So in the short period of time that the astronauts are in space, actually undergo accelerated aging, which then reverts back to normal when they return to Earth. So it gives us an opportunity then to identify molecular signatures that can be used as perhaps as novel targets for drug development that can mitigate or prevent some of these conditions, which can be applied for use on Earth. So the findings can be translated into novel drug development, into novel drugs that can be used for us here. So in terms of the projects, we're looking at immune senescence, we're looking at bone and joint problems, issues with integrity of the blood-brain barrier, looking at infection, primarily in the lung, looking at proteinuria and kidney stone formation, sarcopenia, looking at also cardiovascular deficiencies, and then also some of the GI disturbances. And so this gives us then opportunity to model the systems, which otherwise would have taken longer to create these models here on Earth, perhaps years, but we could essentially model these changes within weeks at the International Space Station by deploying the tissue chips there, being able to then do the analysis and figuring out what those changes are molecularly, and then, as I've mentioned, creating or using that opportunity to create novel targets for drug development. One of the other things that we're interested in would be to vastly improve the technology for organs on chips. So when I talk to the astronauts gone up to space and will be the ones running the experiments, a good number of them are not trained as scientists. They're primarily coming from military background. And so they said, you need to make the system as robust, automated, essentially turnkey technology, or in short, they call it, make it astronaut fighter pilot proof. And so that will obviously poses a large challenge for us as tissue chip developers. So just to give you an example, if we are to conduct an experiment on 24 chips here on Earth, and so this would be essentially looking at 72 samples. What we're looking at would be multiple syringe bombs, 4 incubators, each of which are 48 cubic feet, multiple tubings, large number of collection vials. But of course, it's severely constrained in terms of real estate at the International Space Station. So the requirement would be essentially a 1.6 cubic feet instead of 48 cubic feet on Earth. Everything has to be shrunk in size, be automated, and be able to essentially miniaturize everything down to the size of a shoebox. But this can be done through partnerships with NASA, CASES, and some of the space implementation partners like Space Pharma, STARS, TechShot, Space Tango, and Bioserve in terms of miniaturizing, making this into turnkey technology, which would then facilitate the adoption of this improvement to make this support instrumentation easily used here on Earth, could become then a benchtop instrument that even an undergraduate student will be able to push the button and be able to make it run, which would severely uncomplicate things in terms of getting the experiments running here on Earth. Just to show you that this is actually a reality and has actually gotten great strides. So we had our first launch. This is actually a picture that I took at the Kennedy Space Center, SpaceX 16 launch that contains one of our tissue chips that's studying immunosenescence. And this will be followed by another launch in March, scheduled for March, although things could change depending on the launch scheduling. Again, this will be for SpaceX 17, with 3 of our tissue chip projects going up in 17, and another one scheduled to go up in April at the Wallops flight facility. That would also contain one of our tissue chip projects. Now to tell you a little bit more in terms of what NCATS is planning or is currently supporting in terms of future initiatives and how we're growing this program more. So essentially looking at the developments in tissue chip technology. We're looking at to do clinical trials on chips, and so should that be coming soon. So essentially looking at how these tissue chips can be used for precision medicine, creating individualized you on a chip. We also have the nociception on a chip, pain on a chip, which is an active solicitation. Right now shown here with the RFA TR number 19-003, as well as creating an immune system on a chip. And those are 2 active solicitations right now in terms of how we can continue to grow the program. One of the things that also have come about recently is the ability for these tissue chips to be able to address and provide some solutions to the opioid crisis. As you may know, over 25 million people in the US experience pain, and certainly opioid misuse and addiction is an ongoing crisis in the United States. The administration and Congress has certainly put a lot of focus on this area, since more than 90 people die every day from drug overdose. So this is something that the NIH has focused on, and essentially was given $500 million a year by Congress to look at this crisis. And so the NIH created a new program called HEAL, which stands for Helping to Aid to End Addiction Long Term. And essentially, tissue chips was able to get some money to use tissue chip technology to be able to model pain, opioid misuse, as well as overdose. And so that is the subject of the current solicitation. And as I mentioned, a new program that is forthcoming would be to use tissue chips essentially for clinical trials on chip to inform clinical trial design, essentially to be able to provide information in terms of the optimal way by which clinical studies can be deployed, for example, by identifying the right population stratification or subpopulations that are most responsive to a particular treatment, and be able to inform how those drugs can be marketed and packaged for FDA approval. So in summary, I'd like to summarize that the tissue chips are more physiologically relevant. They essentially constitute the microarchitecture of the organ, provide tissue-tissue interface, precise flow, perfusion and innervation, dynamic biomechanical cues are represented, as well as spatial temporal chemical gradients, and essentially being able to accurately predict human response in this system. And with that, I'd like to end my presentation. And thank you very much for listening in.
Thank you, Dr. Tagle, for that informative presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to remind everyone that this will be available for on-demand through May 2019. A final reminder, Dr. Tagle will follow up with any questions you've submitted via email. That's all for now, and thank you for joining us. We'll see you next time. Bye-bye.